Okay, so today I'll be talking on antibiotics, types, mechanism of the action, and different targets at which they act. So, in my first lecture, we talk about the antibiotics. What are the antibiotics? As you all know that the antibiotics were discovered in 1928 by Alexander Fleming when he was observing the lysis or the degradations of the cell lines of the Staphylococcus aureus when he was identified the area surrounded by the mold, that is Penicillium notatum. So it means the Penicillium notatum, this area, they're showing the lysis of the cell, and this is a Penicillium colonies. All the cells which are around the Penicillium colonies are not growing here. So whereas compared to the other area where the Penicillium colonies are not present, but the colonies, Staphylococcus aureus colonies were growing. That shows the degradation of the cell or the lysis of the cells takes place because of the release of some bioproduct from these antibiotics, from these fungus or mold. So that was an observation which led the Alexander Fleming to think of what is basically inside the Penicillium notatum. And then he discovered the molecule from the Penicillium notatum we called penicillin. That was the first antibiotics in 1928. And the term antibiotics derived from the word antibiosis, meaning the mechanism of inhibition observed between the colonies growing on the solid media. So this is how the antibiotics, the word antibiotic was discovered. Antibiotics are substances produced by the microorganisms that inhibit other microorganisms. So sometime even besides antibiotics, there are some toxins which are produced by one kind of a microorganisms which may inhibit the growth of other microorganisms. So this is kind of a sometime association between these two kind of a microorganisms in different form, like a we call it like a parasitism, symbiosis, mutualism, right? So all these kind of associations between the microorganisms are there. So in this case, one of kind of a microorganisms are inhibiting the other microorganisms. However, in common practice, the synthetically derived antibiotics are also present. So basically, the antibiotics first discovered from the mold, Penicillium notatum, are the microorganisms byproduct, but later on the same antibiotics, the structure of antibiotics were also synthesized through manually or through synthetically. So this we call the synthetic antibiotics. Or these antibiotics may also derived from the these molecule of structure of antibiotics. So we can synthetically modify it, we can derivatize these molecules, and we can also play around to increase the activity of these antibiotics. There's some interesting fact that Penicillium notatum was not an efficient producer of penicillin, number one. Penicillium chrysogenum was identified as the better source of penicillin. Then Streptomyces is a filamentous bacteria, uh, just like a fungus, and that commonly inhabited in solid soil and source of more than half of the antibiotics used today. So this is how these microorganisms are producing several kind of antibiotics. So if you look at this table, you find the gram-positive rods bacteria, Bacillus subtilis, then we have Nemibacillus polymixa. They form a polymyxin, bacitracin. So these are all antibiotics. So antibiotics are produced from Bacillus subtilis, bacitracin. Then we have P. polymixa, polymyxin, which is the latest antibiotics we call polymyxin or colistin antibiotic, which are called as a last resort of antibiotics. Actinomycetes, they have Streptomyces nodosus and amphotericin B is produced, which is antifungal antibiotics. Then we have Streptomyces venezuela, produce the chloramphenicol, and Streptomyces aureophacine. Aureophacine produce the chlorotetracycline and tetracycline derivatives. So different tetracycline and chlorotetracycline derivatives were identified, were isolated from the Streptomyces aureophacine, and then we have a Saccharopolyspora erytheria. And this Saccharopolyspora erythra is erythromycin, which is very common antibiotics used for common cold, throat infection, sore throat, and all. Then Streptomyces griseus is streptomycin, aminoglycoside group of antibiotics. Then Micronospora purpuri, gentamicin. Then fungi. Among the fungi, we have then cephalosporium species like cephalotin. Then we have cephalosporin or cephalothin, or the group of cephalosporin generation of antibiotics. And then Penicillium grisoflavum. These are grisoflavin, which is again one of the common antibiotics, and Penicillium chrysogenum. Penicillium chrysogenum is a penicillin. Penicillium notatum is also used sometime to get these penicillin. So this is the class of this is the different type of antibiotics which are produced from these bacteria and actinomycetes and fungi. Now, the prokaryotic cells are eukaryotic cells, the better antimicrobial agent targets. Both prokaryotics and eukaryotic cells are the best target for these antibiotics. And target here means like you need to have some specific target. Where does the antibiotic act on? It may be membrane, it may be protein synthesis, it may be some enzymes, it may be modification, it may be flux pumps, porins, and so on. So prokaryotic cells are easier to target because they do not resemble the human cell, unlike fungus, protozoan, and helminth, which are has some similarity with the human because they are close to the eukaryotic cells. So many eukaryotic functions are the targets which are present may have some analogy, functional analogy, or to some extent a structure analogy, which may like confuse the antibiotics to target whether these pathogens are the human cells. So that's how these, in case of these human pathogens, human cells are more at risk. The cell wall is different in prokaryotes and this eukaryotes, and the structures of the ribosome is different. Prokaryotic have 70S, and whereas eukaryotes have the 80S ribosomes. And the probability of host cells damage is less. So to act on the prokaryotic cell, the antibiotics have less chance of affecting or side effect on the human cell. In case of prokaryotic antibiotics, they are less prone to be to show the side effect on the human cell, whereas in eukaryotic antibiotics, anti-eukaryotic antibiotics, they are more like chances of having showing the side effect on the human cell because the cell similarities. They have a selective toxicity is higher in case of eukaryotic system. What are the different types of antibiotics? So basically, antibiotics are categorized into two different types. One we call bacteriostatic, another we call bactericidal. So bacteriostatic antibiotics are those which inhibit the growth of antibiotics, right? They don't kill. Whereas the bactericidal is the one which kill the bacteria, right? So the targets which are used in bactericidal are such that they completely kill the bacteria. So bacteria are no more weeds. Whereas in case of bacteriostatic, in the presence of antibiotics, bacteria stop growing, multiplying, and so infection could be stopped or controlled at that point of time. But when the concentration of antibiotics get reduced or eliminated from the system, from the site, then again the bacteria start growing. So we call the bacteriostatic antibiotics are such kind of antibiotics are called bacteriostatic antibiotics. The type of antibiotics based on the spectrum of activity. So what is the spectrum of activity is again important. So antibiotics may have like broad spectrum, they could have a narrow spectrum and extended spectrum. So there are three classes of antibiotics. So narrow spectrum antibiotics like penicillin G, vancomycin, so they can very specifically effective against the gram-positive bacteria, right? Where the broader spectrum could have like tetracycline, carbapenems, fluoroquinolone group of antibiotics. So there are the list of antibiotics under these groups, and they are active both in gram-positive as well as on the gram-negative bacteria. So they have a broadly covering the range of bacteria. Now, extended spectrum is like amoxicillin, clavonate, and then piperacillin, tazobactam. So these are antibiotics which are basically antibiotics with inhibitors. So we have like clavonic acid and tazobactam are linked with these antibiotics, and they show the inhibition of beta-lactamases. So we call them as a extended spectrum beta-lactamases. So they can work on extended spectrum beta-lactamases like different class A, B, C, D beta-lactamases, which we'll discuss in the mechanism part. And they're designed to overcome the resistance and broad coverage of the infection caused by these bacteria producing these antibiotics. Now, types of antibiotics based on their target, mechanism, and action. So again, you can categorize the antibiotics on the basis of their target. Where does it act on? Where does it hit? Where does it target? As well as the mechanism of action. How does it inactivate? How does it stop the growth? Or how does it kill the bacteria? So all these are the called mechanisms. So on the basis of the mechanism, we also categorize. For example, some of the antibiotics, they act on the cell wall synthesis inhibitor. So they are the cell wall inhibitor. So they intervene with the cell wall synthesis and inhibit the bacteria, or they stop the growth of the bacteria by inhibiting the cell wall synthesis like peptidoglycan. For the action is like they block the peptidoglycan cross-linking. So once the peptidoglycan cross-linking is blocked, and nag, nap, nag. So N-acetyl muramic acid, N-acetyl muramic acid, N-acetyl acetic acid, N-acetyl muramic acid. So nag. So nag is being blocked, being stopped, checked at the point. So this is how you block it, because you block the chain. For example, beta-lactam like penicillin, cephalosporin, carbapenems, monobactam, like glycopeptides, vancomycin. These are all class of antibiotics. They are all acting on the cell wall. Now, cell membrane disruption. So disruption of the bacterial membrane leakage. Cell membrane leakage is another action. For example, polymyxin, like eucolistin. Then we have daptomycin. Then protein synthesis inhibitor like 30S, aminoglycosides, tetracycline, 50S, which act on the 50S ribosome during protein synthesis. Then we have macrolides. Then we have lincosamide. Then we have chlorphenicol, and then we have oxazolidone. Halt translation at ribosome. Okay, so at the ribosomal level, when the translation is stopped through either binding with the 30S or 50S subunit in case of bacteria, then such kind of antibiotics called the protein synthesis targeter, protein synthesis inhibitors. Some of the antibiotics, they like fluoroquinolone, ciprofloxacin, levofloxacin, they act on the DNA replication. So they call DNA replication inhibitor. For example, inhibit the DNA gyrase, topoisomerase. So these are the enzymes which are involved in the DNA synthesis during the process of DNA replication. They can be targeted by these antibiotics. So then we have folate metabolism inhibitors. Then these folate metabolism inhibitors like sulfonamides, trimethoprim, they basically block the folic acid synthesis. So the folic acid synthesis is very important to carry on the DNA and RNA synthesis, and so they stop the DNA and RNA synthesis. So when the nucleic acid synthesis is stopped, the cell will kill, our cell will not form, our cell will not continue their growth, or the continuation of the proliferation process is stopped. Now, DNA damaging agents, again, we have a metronidazole. Metronidazole is basically the homologue of the DNA. It causes the free radical production, and these free radical basically damage the DNA strand break. Type of antibiotic classified by chemical structure, beta-lactam, then we have penicillin, amoxicillin, cephalosporin, ceftriaxone, carbapenem, meropenem, monobactam, aztreonam. So this is the class of antibiotics like belong to the beta-lactam antibiotics, and some of the antibiotics are like monobactam, like aztreonam. They are very, very broad spectrum antibiotics. They inhibit the cell wall synthesis by binding the penicillin binding protein. Penicillin binding protein is the protein which is on the membrane and which anchor the antibiotics to act on to the membrane. And now inhibiting the penicillin binding protein can develop a resistance. So if there's a mutation in the penicillin binding protein, it will not bind antibiotics, and so antibiotics start getting resistance. So this is also one of the mechanism of development of resistance. Now, glycopeptides are vancomycin, then teicoplanin, They block the peptidoglycan cross-linking by binding D-alanine and D-L-terminus, right? And aminoglycosides, gentamicin, amikacin, streptomycin, then we have bind 30th subunit of ribosome. So it blocks the translation process by binding the protein synthesis, and then mRNA synthesis will not take place. When the mRNA synthesis is basically misread the mRNA, and so it block the formation of the protein by synthesis of by bringing the tRNA, amino acyl tRNA for a specific amino acid onto the ribosome. So this is how it blocked the protein synthesis. Tetracycline is again doxycycline, minocycline, digocycline. These are all class of tetracycline antibiotic which bind to 30th ribosome. Again, it blocked the tRNA binding process, which is carrier of the amino acid. So when the amino acid will not come, protein synthesis will be stopped. So overall, these antibiotics, they are attacking on the protein synthesis through 30th ribosome, either through misreading of mRNA or through inhibiting the tRNA carrier, tRNA to come to bring the as adapter to bring the amino acid on the chain of the synthesis of the protein in the ribosome. Then macrolides is erythromycin, azithromycin, clarithromycin. These are all group of under the group macrolides, and these bind to the 50th subunit. Again, 50th subunit is important because it blocks the translocation, because 50th subunit of ribosomes, they carries the peptide bond to translocate to the other A to P site, and so this translocation is blocked. When the translocation is blocked, the further amino acid synthesis or the further adapter tRNA should not come, and it will block the the synthesis, the translation of a protein. Then lincosamide is clindamycin. It binds to the 50S ribosomes, inhibit the peptide bond formation. Then we have chlorphenicol. Chlorphenicol, again, it binds to the 50S subunit, peptidyl transferase activity. So binding on 30S or 50S subunit, all different group of antibiotics like tetracycline, macrolides, lincosamide, then chlorphenicol, they all act on 30 or 50S subunit, but they have a different mode of action in inhibiting, even including the aminoglycosides inhibiting. For example, aminoglycoside, they misread the mRNA, where the tetracycline, they block the tRNA binding. Then macrolides, they block the translocation process in 50S subunit, and lincosamide, clindamycin, they inhibit the peptide bond formation. So there's no peptide bond formation, so the amino acid will not link, protein synthesis will be stopped. And chlorphenicol is basically again on 50S subunit, the peptidyl transfer activity is blocked. If you look at the oxalidenone is a linoside and tezolide, tedizolid, sorry, linezolid and tedizolid, linoside and tedizolid. Now, linoside and tedizolid basically prevent the 30S and 50S complex formations inhibition process, and fluoroquinolone is basically ciprofloxacin, levoxycillin, and maxifloxacin, inhibit the DNA gyrase and topoisomerase activity. So this activity is basically involved in the unwinding the DNA during replication process. So they block the replication process of the DNA, and so the cell will not able to divide and to form the new cells at the site of infection. Now, rifampicin is inhibit the RNA polymerase, block the transcription process. So there's no mRNA synthesis. So again, when the no mRNA synthesis, again the protein synthesis will be blocked finally, because that mRNA will not bind to the ribosome. Sulfonamide, for example, sulfamethoxazole, inhibit the dihydrotrivate and synthase folate pathways, where the synthesis of the various bases takes place. And diaminopyrimidine, trimethoprim is very common antibiotics, inhibit the dihydrofolate reductase, like folate pathway again. And then we have nitroimidazole, metronidazole. They produce the free radicals, and free radicals like hydroxyl radicals and celedoxins are produced, and they damage the DNA structures, and so under the anaerobic condition. Polypeptides like polymyxin, colicin, bacitracin, they all disrupt the bacterial membrane. Like polymyxin inhibit the cell wall synthesis, and bacitracin also inhibit the cell wall synthesis. And peptido and then lipopeptides are basically daptomycin insert into the bacterial membrane and depolarizations of leading to cell death. So all these class and group of antibiotics are basically inhibiting the bacterial cell through different mechanism. They're having different targets to that. With this, I think we conclude that use of antibiotics in terms of like mode of action, it is important to understand what are the different targets. Now, once you understand the target, then you can easily identify the molecule which can act on that target to inhibit it. Now, in order to develop a resistance, which we'll discuss in some other next lecture, that if these targets are modified, for example, if the ribosomes, enzymes, gyrases, topoisomerases are some other targets which are basically considered as a target for the antibiotics are modified, are mutated, are changed, right, then these antibiotics will not act on. For example, in case of beta lactamases, if penicillin binding protein is modified, or if penicillin binding protein is like changed or mutated, then the antibiotics will not bind to this penicillin binding protein, and the membrane will not act on the membrane to disrupt. Therefore, this is important to this is important to find the targets before we learn it, and then we can go for the identifying the antibiotics to act on. Okay, thank you very much. And now we have another lecture to talk about the antibiotics mechanism.